So welcome everyone to this 2024 update webinar being hosted by the Australian Fungicide Resistance Extension Network, which you may know as AFREN. I'm Bridget Penner from Ag Communicators, and in today's session we're pleased to welcome SARDI's Tara Garrard and Hari Dadu from Agriculture Victoria. Tara is a cereal pathologist whose research is focused on barley, net form, net blotch, and monitoring the development and movement of fungicide resistance in cereal diseases. Hari is also a cereal pathologist whose research has included developing strategies for the management of foliar diseases of wheat, barley, and oats. Both Tara and Hari will shed light on the topic of net and spot form of net blotches today in this webinar. So just to give you a quick overview of AFREN, so AFREN is a collaborative network of Australian grain stakeholders with a responsibility to develop and deliver regionally specific messages regarding fungicide resistance. So we have a network of plant pathologists, communication experts, and really the aim is to develop communication and extension resources and share these on our website. So some of the things which we've produced include a fungicide resistant management guide, workshops, fact sheets, podcasts, webinars like today, and that sort of thing. So check out the AFREN website when you've got a minute, and these are some of the organisations who are involved in the project. Just a quick note on logistics before I hand over to Tara. If you have a question, please feel free to drop it into the Q&A box. We'll have a Q&A session at the end of the webinar. And if you do have any issues, please put those in there as well, and we'll try to address those as we go through. But for now, I will hand over to Tara. So over to you.
Thanks, Bridget. Hello, everybody, and welcome to today's webinar. Thanks for coming along. It's great to have you here. So we're going to talk about both net and spot form net blotch and the current fungicide resistances we have nationally for those, and some management strategies that can help us to continue to successfully manage disease whilst we have these fungicide resistances, and also to help prevent further development of fungicide resistance. So Bridget spoke a little bit about the AFREN network and the resources that we have available in those. And I'm going to start with this national overview of fungicide resistance in Australia. And this is from the AFREN website, which you can access at any time and see some up-to-date resources. This gives us a snapshot of each state, what we currently have present for all of the cereals, as well as canola and pulses. We're going to go into a little bit more detail today, obviously, on net and spot form net blotch. So a little bit of terminology. We're going to be talking about fungicide resistance, and we have three key terms within that. So when we're referring to resistant, we mean the fungicide is no longer effective. And reduced sensitivity means that we've then got a reduced amounts of sensitivity from that fungicide. Sensitive is indicating that we're still getting good efficacy and that the fungicide is still effective. And you can see in the image here, we've got a petri dish with the fungi growing on fungicide-soaked discs, and that moving from sensitive through to resistant, we get more and more fungal growth. So that's a little bit of terminology that we're going to be using throughout. It can get a little bit confusing because we're often also talking about the varietal disease resistance as well. This is separate, and we're talking about the host resistance of the plant, and it's different to the fungicide resistance. So across our barley diseases, what are the different risks that we have of fungicide resistance developing? You can see here that both net and spot form have a serious threat of fungicide resistance development. Powdery mildew is in the same boat. Scald has a medium level of threat, and in the past we thought that leaf rust would have a very low level of threat of developing disease resistance. The situation that we're in at the moment is we know we do have fungicide resistance that's developed in both net, spot form and powdery mildew. Scald, we don't have any known resistance, but leaf rust, we actually have started seeing reduced sensitivity. So this is kind of the theory that we had in our understanding earlier on in Australia's journey in this, and we're certainly learning a lot more as we go. Today, we're going to dig in a little bit to what some of those factors are that put our net and spot form at a higher risk of disease fungicide resistance development compared to some of our other diseases. So when we break that down, really what we're talking about is the higher level of disease pressure that we have, the higher the chance of fungicide resistance development we have. And our disease pressure is increased by pathogens that have a short latency period, sexual reproduction, and a high spore production. And the theory here is that the more spores that the pathogen has the ability to produce, then the more spores that potentially have mutated containing a resistant mutation, that when we put fungicide out, you can imagine if we've got 1 billion spores and one spore has a mutation that's resistant, the fungicide's likely to remove all of those others, and that will be the last spore standing, enabling that one to reproduce. So net form and spot form net blotch both have relatively short latency periods. We have active sexual recombination in Australia, and they both have the ability to produce a high number of spores. Our fungicides, so when we're using a single mode of action repeatedly, we're putting the same selection pressure on the plant and on that pathogen over and over again, which means if we go back to those billion spores and the last spore standing is the resistant one, then when you come back again, that's the one that's reproduced, and the next application of the same fungicide with that same mutation present is not as likely to be effective. Susceptible varieties, so we know that varieties that have susceptibility to our diseases are going to have more disease that is produced within the growing season compared to, say, a moderately resistant or a resistant variety. Early sowing, and often this is about the number of disease cycles that the pathogen is capable of completing within a growing season. Amenable weather, so we know there's different conditions that are conducive for disease, and close rotations and stubble retention. So we'll now talk a little bit more specifically about how those factors come into play for both net and spot form net blotch. So if we have a look at the life cycle, net blotch and spot form net blotch have a similar life cycle, and the key factors here are that we have survival of the pathogen on stubble at the end of the growing season. So that stubble is then providing initial inoculum that is then moving into the new paddocks in the following growing season. Spores are then being produced on the leaves from the lesions that have developed, which are then able to move across the growing region. So this can move from paddock to paddock and can move within or throughout a region. The spores that are produced off of the leaves do have that ability to go long range, which is a bit different to, say, Septoria triticide blotch in wheat, where you're reliant on rain splash of the spores up the plant. These ones can be picked up in the wind and carried to other paddocks. Those lesions toward the end of the season are then where the sexual stage is then able to develop, and we can get recombination, and then the new cycle starts again from that stubble in the following growing season. So stubble retention and pathogens that are retained on stubble is really a theme that we're seeing with some of the most common diseases in our growing systems now. It is such a big driver because it's providing a high inoculum load season to season. So in terms of net blotch risk factors, growing susceptible varieties with little diversity in the region This means that the pathogen is growing on the same variety, which means it's the same virulence that is then spread across the region. And that's a selection pressure that we're then putting on our pathogen population. Growing barley on barley in a stubble retention system means that we're sowing into a paddock that has a high inoculum load if we've had infection in the year before. It's going to be a lot harder to get on top of disease when we've got that high infection, that high inoculum load there ready to go. Early sowing, so once again, this is about the length of the growing season and the opportunity for the number of disease cycles to occur within a growing season. Wet conditions, both of these diseases will thrive under wetter conditions. Certainly, spot form net blotch has much better tolerance in the lower rainfall zones, and we see that persistent in areas where they don't have high rainfall. Net form net blotch does prefer the higher rainfall, and we see that a lot more in the medium to high rainfall zones. It's the humid weather and those warm conditions that we get in spring that really allow net form net blotch to thrive. So in the winter, when it's a bit colder, that certainly just slows our disease cycles down. And this is some work from Hari, having a look at the amount of yield loss that was associated with the different seasonal rainfalls. So starting at a below average season, you can see the yield losses there are sitting at 0 to 5%. And as we increase into an average, we go up to 5 to 10%. And above average, 10 to 25% yield loss. So really, this is about knowing what conditions you're likely to have within your paddock, what kind of rainfall are you expecting, and how much disease is then likely to proliferate based on that. We want to be making decisions about integrated disease management based on what our potential yield losses are. So I've talked a couple of times about the pathogen population and the diversity that we have within that. And part of this is driven in net form net blotch by the fact that we have sexual recombination that's occurring. We also have growing systems that are very conducive for net form net blotch with stubble retention systems. We've been pathotyping net form net blotch isolates across the nation for a number of years through a GRDC funded project. And what we have in this table here is running down the side isolates that have come from different areas in Australia. So you can see South Australia, Western Australia, and across the top there we have 36 different genotypes, and these make up our differential set. So these different barley varieties have different genetics in them in terms of resistance to net form net blotch. This allows us to have a look at the variation in virulence patterns nationally and keep track on some of our resistance sources and whether or not we've got new virulences that are cropping up. There is quite a lot of diversity here, and you can see by the different colours on the graph that we do have a broad scope of different pathotypes for net form net blotch. You can look at some examples there like maritime, which does have a single major gene for net form net blotch. And for the most part, it is still coming across as a resistant variety. But virulence that on that genotype is around, and we do see that crop up from time to time. So we can see in our Queensland samples here where we're getting 7s that they have virulence on this particular genotype. Really, this is an example of the amount of diversity that we're dealing with. We're not dealing with a single pathotype. We've got a diversity, and this is adding to why it can be quite difficult to manage the disease. But also when we've got fungicide resistance that develops, the role that it plays in the pathogen population and what percentage of the pathogen population contains that resistance. And when we look at a similar graph for spot form net blotch, we actually have a lot less diversity in the spot form net blotch. We see that it is slower to progress, adapt, and change to the selection pressures that we put on it. It still does, but it's not happening at as fast a rate as we're seeing in the net form net blotch. We don't entirely understand why we see that difference between spot form and net form, but we do, and it helps us to then understand how we need to manage these diseases. So we're now going to move on to talking about the current fungicide resistance statuses for these two pathogens in Australia. And we'll start with net form net blotch. So with net form net blotch, we have 3 different modes of action that are registered for control of net form net blotch: the DMIs, the group 3s, the SDHIs, the group 7s, and the group 11s, the QIIs or strobilurins. And in this table, we've got the compounds that are affected in each of these groups. And then we've got the states that are listed here. Now, R stands for resistance, RS stands for reduced sensitivity, and L stands for laboratory detection. So what this is showing us is that in WA, for example, in the DMIs, we have confirmed resistance as well as reduced sensitivity to net form net blotch in the DMIs. Whereas if we compare Victoria, we don't have that resistance, but we have had a laboratory detection as well as reduced sensitivity. So the reduced sensitivity means that we're still able to get some control, and the level of that is going to vary. Whereas resistance means that there is no sensitivity left to that fungicide. So we have those DMI, resistance, reduced sensitivity, and laboratory detections. Those in Queensland, South Australia, Victoria, and WA. The SDHIs, we have resistance present in Queensland, South Australia, Victoria, and Western Australia. We have dual resistance, where we actually have the mutations detected in the same isolate of the pathogen for the tebuconazole group 3 and fluxapyroxad group 7 in South Australia and WA. So that means that the sexual recombination of the pathogen has combined those 2 mutations into one, and we now have mutants that contain both of those resistances in the one isolate. We also have a detection of reduced sensitivity in the strobilurins in South Australia, and we'll talk a little bit more about that in a minute. So the fluxapyroxad, or the SDHI, resistance and reduced sensitivity, first occurred in South Australia on the Yorke Peninsula, and the CCDM team, including Fran and Wes, as well as myself and Hugh Warwork, did quite a lot of monitoring in the first year to understand what prevalence this had across the Yorke Peninsula. In subsequent years, up until about 2021, we've built a bit of a map so that you can see where these resistant isolates have been collected from across South Australia and Victoria. The red here is indicating resistance, and you can see pockets of that present on the Eyre Peninsula, Yorke Peninsula, as well as over in Victoria. And we then have moderate resistance and reduced sensitivity in some other areas. There's also some green present there, showing some that are still isolates that are still sensitive. So it gives us a little bit of a snapshot that we are now seeing this as quite a broad spread thing throughout South Australia, and that we do need to be aware that this resistance could be prevalent within our paddocks. I talked before about pathogen populations and the proportion of a pathogen population which contains the mutation conferring resistance. And this is some more intensive work that Fran and his team did in 2019 to have a look at the pocket on the Yorke Peninsula and how prevalent this resistance was. These pie charts that are here are showing a pathogen population, so a number of samples that are collected from one paddock of netform net blotch, and what percentage of those then are resistant, have reduced sensitivity, or are still sensitive. And you can see that this does vary and change. Down in the southern parts of the Yorke Peninsula around Minlaton, we've got a paddock there that's got 58% of the pathogen population that's resistant, and 29% of that is still sensitive. So what we expect is that as we continue to put selection pressures on the pathogen within the paddock, and this means by application of a fungicide, we're putting a selection pressure on and we're selecting for those resistant mutants and those isolates. If we do this again and again, slowly they're going to build up to a larger proportion of that pathogen population. Now, if we think of this in the sense that 58% of that pathogen population would not respond to the fungicide at all, we've only got 29% that would still respond to the fungicide. This means that we're slowly selecting for more and more, and over time, we're going to see that paddock moving into a field failure scenario where the fungicide is not effective at all. So this is a few years old now, but it demonstrates the importance of the pathogen population within a region and the percentage of the mutant that we've got present. When we had a look at the DMIs, in this case, tebuconazole, you can see that the resistance was a lot higher and much more broadly spread. And we know that the reduced sensitivity for this mutant is quite highly prevalent in some areas. So I spoke before about there being a detection of strobilurin reduced sensitivity. This has actually come from BASF and some work that they did in 2023. I believe that it was a trial site on the Yorke Peninsula in South Australia, where they've sent samples off for analysis of fungicide resistance. And they've come back detecting the F129L mutation, which confers reduced sensitivity to netform net blotch. So a little bit different. This one doesn't confer resistance as far as we know, only reduced sensitivity. It is only from one sample at this stage, but I think it's really important for us to know that this has now occurred in Australia. We do have a risk of losing this last group of this last mode of action that we have available and registered for use in netform net blotch. So it is there. We will need to be keeping an eye on this to have a look at how much further it spreads and understanding what that actually means in the field and what level of control we can expect with this mutation present. So a little bit of a warning there as we now have resistances and reduced sensitivities for netform net blotch in South Australia in all three of those. That sets precedent for Australia that this could be then continued further. So I'm now going to hand over to Harry, and he's going to touch a little bit more on some of the things they've been seeing in Victoria before talking about disease management strategies. Thanks.
Thanks, Tara. Good morning, everyone joining us from the West, and good afternoon, everyone listening to us from the East. Yeah, continue where Tara left on netform of net blotch fungicide resistance. To look into the status more specifically in Victoria, we have seen some levels of resistance popping up since 2021 for netform, particularly to those two groups which Tara was mentioning about, DMIs and SDHIs. Here we have a survey map that we did from 2021 to 23 across Victoria. those red circles which are representing the insensitive populations. It can be either reduced sensitivity or resistance for both DMIs or SDHIs, and the blue circles represent the sensitive populations. So what you can see is the majority of those pointers show that there is increased amount or increased incidence of insensitivity towards fungicides within net blotch populations in Victoria. So this means that there is high risk, as we have found in South Australia, in controlling net form net blotch using fungicides. So more specifically, if you look at the numbers that what Tara was mentioning about in South Australia, we had some analysis done for the samples collected in 2023 from CCDM, where they have found increased proportion of resistant populations for both DMIs and SDHI group of fungicides. As you can see on resistant populations, there is 41% of populations are deemed to be resistant for DMIs, whereas for SDHIs it is 22%. I had have a look at similar numbers for 2021 analysis, where we had only 19% of populations which are resistant towards DMIs, but there were none for the SDHIs. So this shows that in two seasons we had double the population sizes which are showing resistance for both the fungicides in net form net blotch. So this is on the back of increased use of susceptible cultivars in the region. And also we had really good wet seasons, which means there is more disease on the susceptible varieties. And again, there is requirement of multiple applications from different fungicides throughout the season. So which means there's selection pressure on the resistant populations, and they have utilized the opportunity and grown in presence. And also this picture also shows that there is sensitive populations of about 54% for DMIs, and also for SDHIs, 41% of populations are still sensitive. So which means these fungicides can still be used and maybe partially effective in regions where those sensitive populations are available, but make sure that in coming seasons we'll see reduced performance from all these groups for net form. And what this means actually in a trial or in your paddock. So we have we've been conducting some trials on fungicide strategies for net form and spot form for close to a decade now, which is mainly done by one of my colleagues, Mark McLean. So what he found in all those research days was Systiva was proven to be best effective treatment, as you can see in this graph on the blue markers, which is representing the data coming from until 2021, and followed by foliar applications at 31 or 31 plus 39, which are stem elongation and flag leaf emergence, when compared to the untreated control, which had nearly 10% of yield loss until 2020s. But whereas coming to 2023, which is coming from a trial that we conducted at Wallop in Wimmera region, the orange and the data is represented by orange bars. You can see that Systiva performance really reduced there because of increased proportion of fungicide resistant or insensitive populations, and also it is not significant. And also it is not significant when compared to the untreated control. This shows it might happen in large bases as well. So we have had reports coming out of from farmer paddocks where they have seen reduced efficacy of Systiva, and they have to go for multiple applications of other foliar fungicides. So this might be happening, and in coming seasons, if we have more conducive weather conditions, and also if you have had a susceptible or highly susceptible variety, then the chances are that you will see reduced performance from Systiva and also from DMI foliar fungicides. So that's the case of net form of net blotch fungicide resistance in Victoria. And now I want to look into spot form of net blotch fungicide resistance status in Australia quickly. If you look at the table which just Tara presented for net form, it is also a very similar table for spot form. On the top, we have fungicide groups and different compounds and different states where the resistance or reduced sensitivity is present, and what are the industry implications for each of those categories. So for spot form, in comparison with net form, fungicides are still quite good to control. because there's not many, because most of the major parts of Australia show good efficacy levels. But whereas CCDM research has shown that in Western Australia and in Queensland particularly, the fungicides have been shown to have reduced efficacies, especially group 3 and group 7, which are DMIs and SDHI specifically. In these two states, we have identified mutations for resistance and also reduced sensitivity, which means you'll have partial to incomplete control for spot form. And for the other parts of the country, we have detected reduced sensitivity in South Australia and also even in Victoria. So this means you might see reduced efficacies in these two states, but it is quite insignificant when you compare with what is happening in Western Australia and Queensland. In terms of quinols, which is group 11, Tara mentioned that there is significant reduced sensitivity in net form of net blotch, but when it comes to spot form, we haven't detected any resistance or reduced sensitivity yet in across the country. So this means those group of fungicides we can still use and protect crops from spot form. That is about quickly on spot form of net blotch. But if you look at the reasons why we have these resistance issues, it is once again use of susceptible varieties and also really good conditions for the disease, and also which means there's more applications of fungicides and then putting stress on the populations which produce more variants which can have reduced sensitivity or capabilities of resistance towards the fungicides. So now we have the risks that we have from spot form and net form. So I'll talk about the strategies that we need to use in terms of variety resistance or fungicide integrated disease management strategies and how we need to use fungicides. Talking about fungicide resistance management, the larger consideration is always need to be given for variety selection, and then followed by integrated disease management strategies, or probably called as non-chemical methods to control the disease. And the least emphasis should be given to fungicides to protect the crops. And now I'll discuss each of the strategy individually with some evidence to show why they are very important. Starting off with variety selection, Tara has clearly mentioned about why we should not be using susceptible varieties, because susceptible varieties will not have resistance genes. This means they are more prone to disease, and in seasons where weather conditions are really conducive, we get more disease on susceptible varieties, and this means you'll have to apply more fungicide applications, and then there's a chance or high risk of fungicide resistance. In the alternation, in the contrary, if you use resistant varieties, they just have more resistance towards the disease, and probably that means less use of fungicides and also less yield loss at the end. And also susceptible varieties will carry disease inoculum from season to season, because net form and spot form are stubble-borne diseases. And to find the ratings for your variety that you're using, we always recommend to use the latest cereal disease guide, probably that you can find it at National Varietal Trials website, or else even you can refer to any state, local state disease guides that are being released by different states. So these latest disease guides are important just because the disease keeps on changing every year with different strains, and which means we get a lot of different reactions on different varieties, and the ratings keep changing each season. So that's why we need to always have a latest cereal disease guide so that you are up to date, and also you know what strategies you want to actually have on that particular variety. So when choosing a variety, if your variety is rated as SVS to PS or very susceptible variety, so we always recommend to have a proactive plan in terms of fungicides. And also if you have a variety which is moderately susceptible rated variety, that means you just need to monitor the variety throughout the season, and based on the conditions that are available, have a plan for fungicides to use, but not necessarily using fungicides straight away. But if you have a moderately resistant or high resistant varieties in your paddock, that means you have very limited reasons to use a fungicide. And also these varieties need to be monitored for any changes in the pathogen during the season, so that if you have to use a fungicide, you may use, but in our experience, in 9 out of 10 seasons, you don't need to worry about a fungicide for if you have a variety which is highly resistant. I also have some evidence to show why we should not be using susceptible variety. For spot form, we have conducted decade-long research trials in field in Wimmera and Mali, and also across the country. So what we have found for spot form is spot form yield loss varies with the varietal resistance and also with the grain yield potential for that region. So if your grain yield potential is more than 2 tons per hectare, then we have seen yield losses happening. But if it is below 2 tons per hectare, then we have not seen yield losses, irrespective of how much disease you have in the paddock. The yield losses can be greater with highly susceptible variety. In all those experiments, what we have found is anything up to 35% is possible for susceptible varieties for spot form. But if you're talking about other varieted varieties, as you can see from the graph, they are probably much less. And with moderately susceptible, moderately resistant variety, the yield losses will be the lowest. And also you won't need to have a fungicide in place. Whereas if you're talking about net form, so we have conducted similar yield loss field experiments across Victoria. So in 2023, we have found nearly 22% of yield loss when a susceptible variety such as RGT Planet was used. But in contrary, if you replace it with a moderately susceptible variety such as Titan AX, we have found no yield loss. The infection was much less than what we had on RGT Planet, and also there was no yield loss. And on top of that, we didn't need to use a fungicide for controlling the disease on the moderately susceptible variety. So this shows a simple change, a cost-effective change. If you move from one rated variety, worst rated variety, to a better rated variety, you are taking the fungicide usage out of contention. So it is very simple and it is cost-effective. So that's why we say a variety selection has to be given major consideration, because you're starting with a good variety, which means you don't need to really look into fungicide, and also which means reduced your economics as well. Now, the other strategies that I want to talk about is crop rotation. So crop rotation is found another cost-effective strategy to reduce the disease inoculum and also get better out of controlling the disease without using the fungicides. In our research, what we have found is having a break between 2 barley crops for at least nearly 3 years, we have found an incredible amount of reduction in the stubble, and also which is carrying the disease inoculum. If you want to look about different crop options, we recommend to use any of the crops that are other than barley. So more diversity is found to have better results. In a research that we have done in Victoria, so we have found if you are using a susceptible cultivar on a susceptible barley, then we get high amount of inoculum levels, which means there's more disease in the following season. But if you replace with other crops such as oats or brassica or wheat or even chemical fallow, we see the levels of disease reduced significantly, and which means that there is less disease in the subsequent season. Alternatively, if you want to manage the stubble by burning them, we also see a significant reduction in the amount of levels of the disease. At the same time, if you are using a resistant barley on top of a susceptible variety, then you will also see significant reduction of disease in the next season. So that's what we have seen in these crop combination experiments. So we recommend, based on this research, we recommend crop rotation is a good idea, or good strategy to reduce the disease in crop and also reduce the usage of fungicides. In another research that is currently being done at Western Australia by Jason Bradley and his team. So they have found in those situations where you need to have barley to barley because of compulsory reasons, you may use a resistant variety instead of a susceptible variety. What they have found in this research is if they have used a susceptible cultivar on a susceptible cultivar stubble, they have found high amount of disease severity in the following season. But if they have sown any barley variety on maxima stubble, which is actually a moderately susceptible variety for net form or spot form, they have found less amount of disease in the following season. So this shows that if you have to use barley on barley situation, then please use a resistant variety so that at least you can get reduced in terms of disease severity, which also means reduced yield losses and also less need of fungicides. The last resort, as I said, is to use fungicides when we have seasons such as 2022, as we had in Victoria. So it was really wet both in winter and also in the spring, so which caused more disease. And in those situations, you might have to use fungicides. And when you start thinking about fungicides, avoid unnecessary use, because follow these principles that I'll just talk about. So not every blotch of a leaf is considered as a fungal disease. In barleys, we know they are very sensitive to weather conditions and also nutrition types. So because of those discrepancies, we generally get different blotching symptoms on the leaves. So these blotching symptoms, not all the times is a disease. So make sure that you correctly identify the symptoms, get tested or get your advisor opinion, or also get tested at the laboratories so that you know that it is a disease and you might need to take a fungicide. But if it is not a disease, then you don't need to worry about it. Always have a look at the disease levels in your paddock. So the disease levels may not be significantly present. If you want to have a fungicide, they need to be significantly present. Unless the disease levels are high, you're not required to use a fungicide. And again, late epidemics are of less concern. All those beyond flowering stage, if you have more symptoms that are coming up, you just remind yourself that that stage is not a stage where you want to put a fungicide on. The more concern is when you have a rainfall or disease in the early reproductive phase, such as that flag leaf emergence or on the flag leaves, then you need to be using a fungicide. And also we get more benefit when we used fungicides in those stages. And also have a look at the decision support tools to make your decision. For net form on net blotch, there is incredible amount of information that is available on the websites, GIDC websites. And for fungicide resistance management, we have information on AFREN as well, and also refer to fact sheets for each of these diseases to understand what fungicides are being used or what stages you need to target as well. And also there is a NetForm NetBlotch app that is going to be released very soon, so which will help you to make your decisions on fungicide. And when you decide to use a fungicide, as I said, there are critical stages that we consider to be using fungicides. If you find in our research that we have done over the last 10 years, we have found seed treatment, which is Systiva that we have used, plus a foliar fungicide spray at Z37 or flag leaf emergence has been most beneficial. And apart from that, we also found two foliar sprays, one each at stem elongation, that is at Z31 to 32, and also at flag leaf emergence, which is Z37 to 39, proving to be really good in controlling the disease and also in having yield gain. So why these stages are very important? Because these stages are the stages where we get flag leaves emerged, which are actually considered as the yield contributing leaves. So target those stages to get most benefit if you're actually looking to target a disease, which is a net blotch or spot form. Now moving on to other things, when you're using fungicides, as I said, we have the risk at the moment. So what we need to do is only spray if necessary. If it is not necessary, always restrict using fungicides, and also avoid using fungicide actives more than once in a season. Same fungicide active more than once in a season, because if you use same fungicide active, then there is a chance of increase of the amount of or frequency of the populations which are resistant to that particular fungicide active, and which means there is ineffective control from the use of fungicides. Whereas if you use a fungicide, always prefer to use more than one fungicide mode of active in a same combination. So for suppose use group 3 plus group 7 or group 7 plus group 11, so that you get better control, which means you are using two fungicides, which gives extra protection. And again, there is less chance for the fungicide to show more resistance towards two different mode of actives. And also try to avoid using group 7 SDHIs and also group 11 quinol or strobilurin fungicide actives more than once in a season, because these fungicides are considered highly risk fungicides to develop fungicide resistance. So this is happening in septoria nitrate blotch as well, and also it already happened in net form. So we need to be careful about these fungicides. So we generally use them very less in quantity, and that means like we can prolong these actives for more years. So in summary, what Tara and I have spoken about is net blotch of fungicide resistance is becoming more prevalent across the country, and we need to be vigilant about the progress and also understand what are the risks actually to your paddock and what resistance are actually present in your region. So if you have an understanding of what is happening in your region, then it means like you can take actions accordingly. And where those resistant risks are found, we recommend to use integrated disease management strategies with a larger consideration or emphasis on variety selection. Always promote resistant varieties, and where susceptible varieties are being used, try to use non-chemical methods in order to get the control of the disease. That include crop rotation, delayed sowing, as Tara said, and then there is other agronomical adjustments that you can make, such as white sowing or reducing the density of the population, which actually allow reduced humidity within the crop canopy, and that means there is less disease as well. And always act when you're trying to use a fungicide, always act on the conditions that you are in, and also when you're using them, make sure that you're following all the fungicide resistance management principles, and also target key timings for the fungicides, such as flag leaf emergence or stem elongation, those stages. And also if in longer seasons, you're always if you have more disease and if you have very less control, you can use a fungicide spray at Z55, which might give some benefit, but in most of the seasons we have seen really uneconomical to use a third fungicide. And when you're using more than one fungicide in a season, make sure that you're rotating the mode of action as well. So these are the principles that we might need to be follow in order to slow down the fungicide resistance development, and also make sure that we have those fungicides used for a long time. Thanks for that. Over to you, Bridget.
Excellent. Thank you so much, Harry. Really appreciate that that insight, and thank you also to Tara for your insights and expertise on the net blotches. So just a reminder, if you do have any questions, drop them into the Q&A box, and we'll ask Harry and Tara to answer them. I do have one question for you, Tara. You showed a map with the net form, net blotch resistance detections across SA and Victoria. How can we keep up to date with future instances of or detections of resistance? Is that something that we can look for in the future?
Yeah, it is, Bridget. Thank you. My understanding is that CCDM will be developing a visualization tool that enables us to see what's happening within regions from samples that have been received. So this will enable us to look at maps through an app where we can see what the risk of fungicide resistance is for a particular mode of action within a region. This data will come from samples that have been received, but it'll be kind of de-identified and won't be localized to a point where we know exactly where that's come from. It'll be across an area that we know what's happening. So this will be a really great tool that helps us to understand and have real-time knowledge, because there are a lot of samples that get sent in for fungicide resistance testing, and that data and having it all collated and available is not necessarily an easy thing to communicate. So we'll be looking forward to that when that is released, because it will be very handy.
Excellent, thank you. I think we might wrap it up there, but I just want to draw everyone's attention to the chat box. I've just dropped in a link to a short survey. Your feedback is really valuable to us to ensure that we can continue to deliver resources that are relevant to you. Just on behalf of the AFREN team, I just want to thank everybody for taking the time out to join us today on this webinar. Just a reminder to access the AFREN website for a whole heap of resources, fact sheets, videos, podcasts, previous webinars that sort of thing. So thank you, everyone, and we look forward to welcoming you back to another AFREN webinar in the future. Thanks, Tara and Harry, for joining us too.
Thanks, Bridget, and thanks everyone
Bridget.
for coming along.
Thanks everyone as well.